All right, and the final part of Chapter 20 will be on gene targeting and knockout animal models. So the concept of this is now we not just want to create an artificial DNA construct. So these are, so we've seen now how we can create plasmids, we can create PCR products, we can sequence these PCR products. But now we actually want to go to the area of gene editing and gene targeting. And this means that we manipulate the DNA sequence of a particular organism. So this might mean manipulating the sequence of a mouse. This might mean manipulating the sequence of a crop, so like a tomato plant. And our goal is to be able to do this intelligently, so we can choose the type of manipulation that occurs. And so this approach is just going to be, what happens is that each organisms tend to have very specific rules for how this is done. And so we're just going to illustrate this in mouse to start out with, and that will actually be the extent of what we cover. And so we're going to illustrate this creating a knockout mouse. So this is how you would actually knock out a gene in mouse. The way you do that is in part that you design a vector. So this is a repair vector. And this is, you should think back to your work to the work done in Chapter 6 on bacteria, where you incorporated like a endogenous piece of DNA, either through transduction, conjugation, or transformation, and that was able to exchange with the chromosome exchange itself with the chromosome to modify the host chromosome. So the concept of this is very similar. So instead of having DNA taken from a natural source, which would be a bacteria, you construct the DNA that you want to use for this. And so we call this a repair vector. And so this repair vector has been artificially constructed by researchers. And they've artificially constructed this to have a knockout of the target gene. So it has maybe the beginning and the end of the gene, but the majority of this gene has been deleted and it's been replaced by a marker. So this marker allows us to determine whether or not the cell has incorporated and recombined out. So if you can get this source of DNA into the cell, then you can rely on natural recombination processes to replace the target gene with this allele that you design. And so the way you have to do this is that you want to transform ES cells, so embryonic stem cells, with targeting vector and select cells for recombination. So this relies on the fact that we made a large number of advances in cell culture, and so we can actually grow embryonic stem cells in a petri dish. And so you will inject and transform the cells with this DNA, and then you can use the neomycin cassette to actually, so this is something that you can add very similar to antibiotics that will normally kill the cell. And so the cells that manage to take it up and actually recombine will produce this resistant protein that confers resistance to this drug. And so in this way, you're actually able to identify ES cells that contain the transformation that you want. Unfortunately, we do not have ways to take those ES cells and cause them to grow into a mouse. So we can't cause them to turn into a pluripotent cell that would go from one cell all the way to an entire mouse. But instead, what has been discovered is that you can actually inject these ES cells into a blastocyst from a mouse. And so the majority of this mouse DNA is normal wild type DNA, but then you have injected a cell into this that contains your target gene change of interest. And so this injection is done in vitro. So you would have removed this. So this cell blastocyte cyst would be You would not inject in the mother's womb. You would actually inject it outside the mother's womb, and then you would transfer this back into a foster mother in order for it to grow. And so this develops what's called chimeras, and the idea behind chimeras is that some of the cells, so of the billions of cells that are created, some of those cells are going to be descendants of this ES cell that you've injected. And so if you do it, for example, on a gene that targets, if you do it on a gene that's responsible for the black coat color, then what you will observe is chimeric mice. And so these chimeric mice have partially black fur and partially blonde fur. And that's because all of these cells can encode the gene that creates black fur, but this one encodes a gene that caused the blonde fur. And so they're chimeric because the different cells have different genotypes within it. So it's not like a normal human or a normal mouse where you assume that every single cell has the same DNA content. These are chimeras because different ones have different DNA content. And so after that, you have to breed the chimeric mouse back to the black mouse. And your goal is to identify mice where you can actually propagate it in the subsequent generations. And so for that to occur, the descendants of this ES cell must form the gametes. So if the descendants of these cells are able to form the gametes, then in that case, this can propagate on to the next generation. And as a result, you'll have fully tan mice because they're now no longer chimeric. So these mice are no longer chimeric. And if you interbreed them again, you can get homozygous mouse for your knockout. You can also introduce different types of vectors into cells as well. So if you can get the vector, the DNA plasmid, into the cell via electroporation, they can also use the endogenous enzyme recombinase to catalyze homologous recombination. So this is a picture of how you would actually inject. This is one of the ways that you can inject DNA into the cell. And so this is a cell where you've actually used this fine needle, and this needle goes into the nucleus, and then you'll actually inject a solution of DNA into the cell. So this kind of holds it in place, and then this needle punctures it and gets it into the nucleus. You don't just have to do knockouts. So any sort of promoter that you can get in there typically works. So any sort of construct that you can get to integrate. And so this also allows us to do things like conditional knockouts. So a conditional knockout is using this Cre-Lox system as an example of how this works. And so the idea behind the Cre-Lox is that you have two LOXP sites. So these flank the gene of interest. So this gene expresses normally, and it just has these small, like 40 base pair sequences. And normally the gene expresses completely normal. So if you were able to make this mouse, it would be essentially wild type. But then you can also add a construct that contains this enzyme that promotes expression of this enzyme called Cre-recombinase. And so if the Cre-recombinase is expressed in the same cell as this LOXP, what it does is it actually causes the deletion, the recombination between these LOXP sites. So it causes this looping that actually removes this whole sequence in between the LOXP sites out. And so this allows you to do tissue-specific expression or tissue-specific removal of a gene. So let's say you wanted to remove a gene from the brain and see what the effects of this gene if it's not present in the brain versus something like skin or muscle. And so if you have a promoter that is only expressed in the brain, then you're only expressing Cre-recombinase there, and those are the only cells that undergo this recombination. And then finally, you can also create transgenic animals that express or overexpress a particular gene of interest. So the vector with transgene undergoes homologous recombination. It integrates into the host genome. And then you can recognize this again by using marker genes. And so an example of this is actually GFP. So in this case, these mice have been injected with a construct that promotes the expression of green fluorescent protein. This is a protein that was isolated from jellyfish, but it causes animals to glow green if you bathe them in a specific colored light. So if you bathe them in blue light, they will absorb that light and re-emit it in the green wavelength. And so this is the reason that some of these mice are actually glowing green, is because they've integrated a construct into their genome that actually allows them to produce this protein. So that is a summary of the Chapter 20. So that is the end of Chapter 20. So we've covered a lot of material, but a lot of this is really important fundamental stuff to understanding genetics and understanding how we use genetics in order to understand very fundamental processes of life. The next Chapter 22, we'll talk more about how these actually are used for technology or for human health related issues.